couple people. Yeah.
They're on their way out. And well, anyway, chop, chop. Yeah, it's also cooling down.
As soon as I tell you what I have, and I even text you, and then goes back, you didn't respond me back, so.
All right. I'm very excited to have Molly Lukavich here. Did I get that right? You did. She is we're gonna go big and far and wide, right? She is a research professor and the director of the Large Pelagic Research Center, which started at UNH, all you Wildcats in the room, and is now at UMass Boston. And she spends a lot of time between Gloucester and Hawaii and oceans in between, chasing lots of big animals and learning how and where they go, and including sharks and sea turtles and tuna that we're going to hear about today. She works a lot with fishermen and does that overlap between fisheries and research and engages a lot of fishermen in her research. A lot of high-tech tracking equipment. How do you find these big animals all over the world? So we're going to hear some neat things about tuna. I'm really happy to have Molly here today. Everybody help me welcome her.
Hello, everybody, and I have to say I'm thrilled to be here. There's some old friends and colleagues here that I didn't know were here. And Jennifer and I met a bunch of years ago. I gave a rock talk maybe 10 years ago, and so I'm really, really happy to meet you all, and what a great place, and I hope you all have brilliant careers. When you get to work out of a place like this, it's just an incredible start, so good for you all. And so, if you don't mind, my laptop, by the way, just crashed the screen. And so, always back up. I'm sure you can come to an island and find that your computer is not going to be happy. So I just learned a big lesson that backing up is hugely important. So thanks to Jen for backing me up. All right, so I'm going to talk tonight about, it's not going to be straight, biology, physiology, ecology. When you get to my age, as a woman senior scientist, you might have had some experience. And so what I'd like to bring for your consuming and to think about is how different aspects of a career in science and fisheries and working with humans and fishermen can come together. And I want to give you a taste of what it's like to have taken on scientific questions and to pursue them and to try to study a big, highly migratory animal like tuna, and what are some of the joys and the challenges of work in this kind of field. Okay? So it's a little bit of a personal perspective, right, rather than a straight class. And we'll start with, if I'm in your way, yell. And my title is, I'll go back, Tangled Up in Bluefin, as I said, the different aspects of this life. So let's talk about bluefin. Bluefin tuna are unique because they're warm-bodied, they lived a long time, they go very deep in the ocean. I hate to call them divers because they're not air breathers, but we have depth records to 1,200 meters. They're found from 0 to almost 32 degrees centigrade, so they have a huge thermal range. They're very fast-growing. They have a very high metabolic rate for the bony fishes, the teleos. They're really a souped-up metabolism. And what's unique about bluefin tuna is, unlike a lot of all the other fishes, except for poor-billed sharks and maybe a few others, they warm their bodies. They're up to 18 degrees or so. Doug Fudge is an expert in this field. Warmer than the ocean that they're found in. They warm their guts to speed up digestion and to process their food very quickly. And which Doug has done a beautiful anatomical work on. And they warm their eyes. So they have unique sensory capabilities. Their growth, everything about bluefin tuna, as far as the fish, is pretty extraordinary. And of course, they're very valuable. So just a couple other views. There are other tunas that are big. This is a big-eyed tuna. That's a temperate tuna. And there's another tuna that's a yellowfin tuna that we're about to put a satellite tag on off the island of Hawaii. And so you can all see they sort of look alike, but bluefin has unique characteristics that are not shared by these other tunas. But they're all tasty. That is swordfish, also considered in what we call the highly migratory species, the large pelagic, and also a commercial fishery in both oceans, in the Pacific and the Atlantic. And so where did we start? Bluefin tuna happens to be a schooling species, and if you're a spotter pilot or you're flying in a plane in the Gulf of Maine during summer and fall, you might see these most beautiful aggregations from the air. So bluefin school in aggregations ranging from a few fish to upwards of thousands of individuals. And that's where I got into bluefin tuna. And the other thing about bluefin is that they have this beautiful shape. It's a torpedo shape. And believe it or not, for a long time, the U.S. Navy has been using the bluefin body shape and using it to design basically vehicles. And this is a robo-tuna that was designed by graduate students in the robotics and engineering department at MIT. And robo-tuna has been a staple for, let's say, defense and other ocean sensing things, and is a really, basically again, shows the optimal design of the bluefin shape and how it inspires. Bluefin tuna, if you live in New England, you know there has been a traditional commercial fishery. This is Gloucester Harbor, and so, of course, it's a very valuable fish. If you like sushi, people like sushi, anybody? So you know that bluefin tuna is the most expensive sushi for the most part of all the tunas. And so there have been fisheries for tuna for a very long time, and they support very delicious food, high quality food and protein that you find in sushi markets around the world. And so, by the way, I'm going to point this out just because I'm going to talk about it in a little bit, but right there, those are the fin rays that hold up the second dorsal fin. They're called the pterygophores. So just remember where they are, because we can talk about them in a little bit. So anyway, bluefin is very valuable, and the price to the fishermen at the dock when they come in can range anywhere from $1 or $2 a pound upwards, at the very highest to $74 per pound. It's a rare price, but the point is it commands good prices and high value for fisheries. So bluefin tuna, just like all the other fish species, as far as our interest and what we need to learn about them, are all the basic fisheries questions. All right, and I'm sure you're dealing with this in your learning about fisheries. Where, when, and how often do these species like the large pelagic, where do they spawn? Where are they foraging? How do they get to these locations and back? what defines their habitat, their ecosystems, and for fisheries managers, and for understanding sustainability, we have to know how many are there. And if these species range across the Atlantic and across the Pacific and back, you can imagine how difficult it is to assess the population. So the same questions that drove historical workers like Pete Wilson there are the questions that drive us, who drove my students, postdocs, and others. These are things we want to find out and need to find out. So the tunas, the sharks, and the billfish fishes, their stock assessments are based on population models and statistical models that describe their population trajectories, otherwise known as stock assessment. And these stock trajectory predictions include biological assumptions, the basic. What's their age maturity? How long do they live? And basically, what's mortality rates? What's natural mortality? What are fishing mortality rates? So it's important that these very basic biological realities are known accurately, because of course you need good information to go in, to have good information in your stock assessment, and to be realistic. So for bluefin tuna, the challenge has been that because they range across the oceans, and they're very hard to track while they're offshore, a real question is, are the assumptions used in management accurate? There we go. So who's in charge of the tunas and the biology and assessment in the Atlantic? It's the International Commission for the Conservation of Atlantic Tunas. It's known as RFMO, or Regional Fisheries Management Organization. It's international, and there's two levels of it. There's the stock assessment scientists from the different nations that are party to ICCAT that conduct stock assessments about how many tunas there are. And then you have, on the policy side, the federal managers and the policy advocates and politicians. And so these are two kinds of processes that are involved that come to the table and deliver the science, and they will deliver the management advice and quotas for species like bluefin tuna. Okay, so what I'm going to show you is the basis for the management of Atlantic bluefin tuna in the Atlantic. And I want you to remember this figure, because the science that we're going to talk about is going to be, think about it relative to these assumptions. Since 1981, it's been assumed that bluefin tuna consists of two stocks in the Atlantic, an eastern stock and a western stock. It's assumed that each of these stocks has separate and exclusive spawning grounds. The eastern stock in the Mediterranean, there you go, right there, and the western Atlantic stock is supposed to spawn only in the Gulf of Mexico. And there's a 45-degree west longitude management line, and the assumption has been that the degree of mixing from these two stocks is very low. But what you see in the orange lines, those are migration patterns that have been detected by spaghetti tags, simple ID tags. So we knew for decades that bluefin tuna crossed the Atlantic and even the equator, but still the assumptions in management were that that was very, very low levels, and that was insignificant. So what that means is that tunas, bluefins on each side of the line are managed separately and differently, and that what happened on this side, if there's overfishing, doesn't affect what happens in the Western Atlantic. And that's still the basis for management. All right, so how is that working? Well, if you have any fishermen friends, you know that for a very long time, our local fleets, our commercial fleets, and our recreational fleets have contested many of the assumptions that are used for management for Atlantic bluefin tuna. And this is when the New England Aquarium, in fact, Amy Knowlton, who was just here, was my old office mate. Back in 1993, bluefin fishermen and their spotter pilots came to the right whale group of the New England Aquarium and they said, you do aerial surveys for right whales. Can you come in as the neutral or scientific arbiter of how many bluefin we see at the surface in schools in the Gulf of Maine? Because the government doesn't believe us. In 1992, the assumption under stock assessment was that there were only 7,000 adult bluefin tuna left in the Western Atlantic, the spawning stock biomass, plus or minus the error. And for that reason, there was a recommendation for a 50% quota reduction for the Western Atlantic. And that would have really brought down catches and really affected our New England fisheries. But fishermen said, we see that many fish in one school or two schools in the Gulf of Maine alone, and that they absolutely rejected the idea that there were that few bluefin tuna, but nobody would listen to them. Scott Krause, the right whale director, said, sure, we can work with the spotter planes and try to independently at least document what was at the surface. That was in 1993, and that's where we started the Bluefin Tuna Research Program. That was separate from the government stock assessments, and that was separate from the ICAT process. So this was the first fishery independent scientists that in a while that came to try to figure out could they get better information on schools. And so the protest by fishermen was for better science. That's what they wanted, okay? So as soon as we started, 1993, 1994, this is the basis for asking questions. What do we really know about bluefin? In the first year alone, we had laptops, we had GPSs and cameras, and we equipped 13 spotter pilots with a data collection system, and we counted upwards of hundreds to over 5,000 surface giant bluefin tuna. And that was the beginning of the information that led us to realize that what we didn't know about bluefin tuna, and that it was very, very likely that the population assessment was not accurate. But again, no one uses these methods to assess tuna. For the most part, the population is assessed with fishery-dependent information from catches. So 10 out of the 11 indices of abundance for bluefin tuna in the Atlantic are fishery-dependent catch statistics. So that's where we started. And this is just a taste of the kinds of data that we've been bringing to the table besides catch statistics. So actually, Bob Schipp, a friend and colleague, and maybe some people here, worked with us. What you're looking at in the Gulf of Maine, I know you all know where you are, these are locations of bluefin schools spotted, photographed by the spotter pilot. What you can see is we have real-time information on where bluefin schools were. And that was the first, because you can take these school locations, you can show the sizes of the school, you can overlay that information on oceanographic information, sea surface temperature, the proximity to herring schools, and so on and so forth. So by getting real-time fishery-independent information, it starts opening up our whole view of the ecology, the biophysical size of bluefin tuna life. So over the first years, we were interested in doing, let's call them ground truthing or sea truthing of bluefin schools. And we were also interested in how quickly do these schools travel, because you needed to know, are you double counting if your planes are flying from, say, the Isle Shoals down to Cape Cod? So we needed to know how quickly do they move and when are they available at the surface. So over the years, we went from sonic tracking, which you might have heard about, tracking sharks or tracking other animals, to realizing, okay, we can get local movements, but what about their long-range movements? Can we track animals over a much longer distance and time? And of course, what I pointed out in the beginning is that bluefin tuna are known to cross the Atlantic as early as age year 1. So we needed a tool that would let us track these fish. And so you're looking at, we worked with fishermen over the years to develop methods of bringing a very large bluefin tuna alongside the boat and getting what we call the pop-up cell, like archival tag, into the base of that second dorsal fin. This is a computer. It's got sensors, pressure sensor, a temperature sensor, and light sensor. And we use the computer, the sensors log information on the computer. It's found in the body of this tag. They're $4,000 a piece. These tags are programmed. We program them to remain attached for a year and collect data for a year. So they're meant to, the tag is positively buoyant because of the float. It's good to 2,000 meters. It's meant to swim along the back of the tuna, and at the mission time, the computer sends a charge to the nose cone. There's an 18-gauge wire in there. The nose cone breaks, this ID tag is left in the body of the fish. The positively buoyant data logger pops up to the surface and then starts transmitting data to receivers on orbiting satellites. There are NOAA satellites right now. The data is distributed to us via email. We pay a company, Service Argos, to send us the data. So it's an entirely fishery-independent data collection method that we've been using and pioneering since 1997. So we're still stuck with 1997 technology, people. We need your minds to help us get past this big, clunky $4,000 non-reliable tag, by the way. So that's my pitch for your engagement to take us to the next level of technology. So we've been sharing these methods, and these tags have been put out, and if you've heard talks from other people, these are not air breathers, so they don't come up. They're not GPS tags. They, for the most part, don't have the capability to use, say, a shark tag on tunas or billfish, because they're just not at the surface long enough. So I'll send this around so you can have a look at it. And we use light-based geolocation, which is basically using astronomical methods to estimate latitude and longitude. If there were more time, I would go into that. But, sorry. Here's another look at the tag that you'll have in your fish. And as I said, at each point on the Earth, the time of sunrise and sunset is known. It can basically help you geolocate. You can derive latitude and longitude, an estimate based on day length, and with an accurate clock time. And then we also use additional information to ensemble common filter approach. Basically, you take all the information you can, whether you're at the sea surface temperature, and try to figure out the best track. So that's the nature of light-based geolocation. The tag is not meant to be recovered. It's meant to be expendable, but fishermen sometimes catch a fish with the tag still in it. And if you get that tag back, you get even more data because the tags actually log every minute. And so it's very valuable. And so you want to get word out to the fleets that these tags are out on your animals. And that's how engaged fishermen on almost every aspect of the work. And they're very happy to see the data. Now, I could probably spend, of course, talking to you about all the things we've learned in our cooperative research program with fishermen on bluefin physiology, ecology. But since we only have 45 minutes, I'm going to give you a taste of what we learned using that technology and how it sort of upset the apple cart in terms of assumptions of what bluefin tuna do. All right? So here you see, these are just 4 year-long tracks that'll give you an example of how the very first data returns from pop-up tags showed us that the assumptions about what bluefin do were incorrect, and they're only partially correct. Here's the paradigm. If you remember when I said bluefin spawn in the Gulf of Mexico. Here's the Gulf of Mexico, and in the summer and fall, bluefin are known to come up to these areas, New England, Canada, all the way up to almost the Arctic region to feed. And then they fatten up, and that's where they're the most valuable for fisheries. And then they presumably go back to their spawning grounds in tropical areas where their larvae need appropriate oceanographic conditions. And then they return, supposedly back up here, to feed again. So remember that bluefin are all supposed to go to the Gulf of Mexico. The colors here show you the month of the data, the geolocation. And you probably can't see the gray area, but that's the geolocation error, that error that's associated with light-based geolocation. So you can see that year after year, as we deployed anywhere from 20 to 60 tags per season on bluefin tuna in the New England or the Canadian region, that only about 20 to 25, 30% of the fish in our sample did what they were supposed to do. And the rest went to different places. And you can see that, including the Bahamas, some were just off the Carolinas, some went over to the Eastern Atlantic and came back. And what was interesting is, wait a minute, these are supposed to be sexually mature fish that are spawning only in the Gulf of Mexico. So what's going on? So year after year, the majority of the bluefin that we tagged weren't in the known spawning area. Okay, so what does that mean? Okay, let's look at the kinds of data that you get. Sorry for the size here, but here's the depth profile of this year-long track of fish released from the Gulf of St. Lawrence. And you winter months. The warm colors are the summer months. So you can see that in the summer, when fish are often up here, that fish was over by the Azores. But yet, nonetheless, came back to the Bahamas and returned to the northwest Atlantic shelf the following year. And here's the temperature. You can see the really colder, that's 5 degrees centigrade. And you can see here the temperature at depth. So that's the Gulf Stream signal. When the tuna hits the Gulf Stream, you see the really warm water. And so, in other words, we've been able to show over the course of the year the bluefin tuna's oceanographic associations, its migration path, and basically the habitat that it occupied. And again, if you assume, as we do, that a very fit, fecund, sexually mature bluefin is going to spawn every year, then you have to ask, so where are these fish spawning? They're not all spawning in the exclusive and separate spawning ground in the Gulf of Mexico. So as we were developing the database over the years, we and others of bluefin tuna, what we basically first started putting out there, wait a minute, we're getting results that aren't compatible with the biological assumptions used for the last 25 years in management. So what's up with that? So it's questioning the population structure paradigm underlying bluefin management. And so over the years, my UNH students and postdocs and I and our collaborators all over the place were basically re-examining the assumptions and say, are they lining up with the status quo? There's Rachel asking us, how does it all fit? And the answer is no. My grad students and postdocs and our collaborators spent a lot of their PhDs on reproductive ecology, physiology, predator-prey relationships, their foraging ecology. We were basically looking at what's the basics, what are the basics of bluefin in the area and throughout their habitat, and putting together the whole story of life history and ecology that we think is the most basic, the first floor of fisheries science, that if you have to know the basics, you have to know spawning, you have to know reproductive periods, you have to know larval development areas, if you want to have good stock assessments. And so we put together these stories in, of course, peer-reviewed periodicals, and we presented it to the science public and fisheries managers and conservation advocates. And believe me, it didn't go over well. And the first problem was that bluefin tuna in the Western Atlantic are supposed to have a different age of maturity than bluefin in the Eastern Atlantic and the Med. In the Eastern Atlantic, the age of maturity has been well established for decades as between 3 and 5 years of age. Rather interestingly, in the Western Atlantic, because only big old fish were found in the Gulf of Mexico for the most part, the age of maturity there has been estimated to be much older, even as late as 12 to 14. And that's what National Geographic recently reported as the age of maturity for Western bluefin. And we were able to show through 2 PhDs, thesis work and collaborators' work, that that's not correct. And so we keep at trying to understand where our spawning grounds, because these are highly migratory species, and the tag, the pop-up tag that we sent around, does not detect spawning. We can only infer from the animal's movements and its behavior and the depth and temperature that it might be spawning. The tag doesn't tell you if it's spawning. So what you have to do is get out there and find either the ovaries, the gonads, and see what condition they're in, or using other methods. And so that just shows you, that's a right, that's an ovary, that's in spawning condition of a fish that was sampled outside of the Gulf in the Eastern Bahamas that was in spawning condition. So it's pretty hard to miss in this fish, you might see. All right? So for these long-lived, highly migratory animals, a year-long migration across the Atlantic or the Pacific was really hard to study, let's face it. And a $4,000 tag and tagging, say, a couple hundred years, only a drop in the bucket. But what we can go back to are first principles that you're learning in your classes. First principles of biology, ecology, natural selection, life history. And what we know is it's all about energy in bluefin. that they have to have a lot of food to support their high metabolic rates and their fast growth rates. They go to the highest latitudes in the coldest water of all the tunas. There's a cost for that, they're warm-bodied, and they cover very large distances. So what happens is they fatten up, they come up to New England, they're eating herring, mackerel, squid, they're getting really fat, between 5 to 7% you think of their body mass per day while they're up here. And meanwhile, they're not just fattening up their muscle, they put fat in the muscle over the season, but they're also putting fat around their gonads. And among all the tunas, bluefin are particularly unique, I think, in having a big fat pad by the end of their foraging season up here. By the end of October, if they're ready to leave, this ovary is very, very tiny, and that fat pad can weigh up to 20 pounds on each ovary. And that fat, those lipids, are used directly. They're very quickly taken across this really cool reed duct that probably the lipids are used to make the eggs and sperm for spawning. And it's a really cool system that hasn't been studied very well, if at all, that I know of. Anyway, it's also out there for you people. Okay, what's the answer? Oh, all these the tunas in your sample, they mustn't be spawning. If they didn't go to the presumed spawning ground, well, then they're not spawning. That's what the alternative view said. And wait a minute. The other thing is, based on first principles in life history, if bluefin, if fish delay maturity, then they should be growing faster, because that's the whole point of delaying maturity, to grow bigger, get bigger faster. But what we see is that eastern and western bluefin are growing at the same rate. In fact, they're eating the same foods and they're eating in the same locations. So we could not find anything that would distinguish the ecology of the life history other than specific location of an eastern or a western bluefin. And in fact, a bluefin is a bluefin. And so this idea that these western bluefin were likely to be vastly different, reproductive schedules and age and maturity from eastern kind of defied logic. But that was the assumption for decades. And it was only challenged by Eastern scientists, Europe scientists, and Japanese. But yet, it remained unchallenged in the US until we put the new evidence on the table. And it's still challenged today, just so you know. So, the problem is that bluefin tuna go into spawning condition very rapidly. And once they're done, they have very little evidence that they've been spawning. And that probably is related to their metabolic rates and their life history. So, if a bluefin is done spawning, and if you use conventional methods of histology, staining, you're very unlikely to see any evidence of spawning after they're done, because all evidence is absorbed for the most part of the post-ovulatory follicles within 2 days. So if you look at a tuna weeks after it spawned, you may not see any evidence that it had spawned. And that's been the problem. But the solution is, all right, let's use other techniques of gonadotropin or hormonal analysis, using modern techniques that were pioneered by aquaculture specialists and are now considered the classic way. My former UNH grad student, Gilad Heinisch, who's now running a fish farm in Israel and is still a research scientist, he basically used techniques developed by Hanna Rosenfeld in Eilat, in the National University in Eilat, and took out the pituitary of bluefin tuna and subjected them to hormonal analysis. And like all other vertebrates, their reproductive hormones are as our own follicle-stimulating hormone, luteinizing hormone. Basically, Gilad's work was to profile what we call the GBP axis, the gonad-brain-pituitary axis, which defines age and maturity and your reproductive condition, right? So, not to go into the details, but this axis is very well studied in other species, and Hanna's lab was applying it to the tunas and to bluefin tuna. And what we're able to show with this work, and I'll show you the evidence that young, smaller bluefin in the Western Atlantic had the same reproductive hormonal profiles as in the Eastern Atlantic and in Med. And that's with gonadotropins. There's luteinizing hormone and GSH. And here you see on the right this pituitary follicle-stimulating hormone and luteinizing hormone. This ratio can tell you a lot about the maturity, sexual maturity of fish. And immature fish have these ratios that are much greater than 1 in 10. And mature fish have these very small ratios. And 185 centimeters was supposed to be the age and size of maturity. And what Gillette found when he looked at the smaller fish was that the fish that we were considered sexually immature based on conventional methods had the, for the most part, identical profiles or similar profiles as well-known sexually mature fish. And in fact, you'll see the sample sizes here, but additional work has increased the sample size a lot. So we were able to verify that hormonally, these fish, these smaller size fish, are sexually mature based on their hormonal profiles, and in fact are consistent with fish in the other parts of the Atlantic, the eastern Atlantic and the So, coming back to the tracks and migration, remember I asked, well, if 3/4 of our sample size over the years of annual migrations are not in the known spawning areas, and we make the assumption that a fit, a reproductively capable and a very physically fit tuna will spawn based on life history theory, actually worked on by Eric Chapman, that these fish are spawning somewhere. And there's two ways to verify it. Find the smoking gun, or smoking ovary or testis, mostly the ovary, or find the larvae. The problem is that Atlantic spawning areas that we were predicting were probably out there finding the bluefin. It's a needle in a haystack, because are you going to have larval cruises over the entire Atlantic? No. That's why no one thought it was very feasible to go out there and look, except in known spawning areas. But you could use modeling approaches. This is actually called Cephodine. It's a multi-trophic biophysical model that was developed by our friend and colleague Patrick Lahode for the Pacific and the Indian Ocean. And what the areas lighting up are suitable larval habitat areas predicted by the Cephodine model for bluefin tuna based on the characteristics of larval development. And you can see the med, the known spawning area, heating up, and you see at some point, that's by month. We can't see the month quite well up there. You'll see the Gulf of Mexico heating up as far as suitability. But also note the other locations that look like they could be good places for larval development. And not only that, including off of Africa. And remember that this is supposed to be the only spawning ground, as is the med. So that's all we had to begin with, besides life history, hormonal analysis, but no smoking gun. And based on that, everyone said, well, we're going to ignore this until you have the final evidence. Let's just move on. So in 2010, we got a very nice phone call. Our colleagues at the Northeast Fisheries Science Center, David Richardson and John here, said, hey, we have something that you've been looking for a very long time. And so we met in Hawaii, actually, and they were the first to tell us that they had found the bluefin larvae in the locations that we've been predicting for the last 15 years. And they weren't even looking for them, in fact, as far as tuna cruises. They were found by diverting the foraging samples for whale cruises. So these were NOAA cruises that went out looking for zooplankton for the food base for whales. And no one else had ever been able to look at these samples except the Southeast Fisheries Science Center, that I was always looking in the Gulf of Mexico for bluefin larvae. And so they said, oh, we have your larvae. They were sent to independent labs. There was a double blind on the species they were, and we published this paper in PNAS in 2011. And not only that, but this paper said one of our assumptions, looking at all the data, was that we predicted that based on what we felt was the extent of spawning and the age classes contributing to the spawning, that the Gulf of Mexico might only hold 20 to 30% of Western Atlantic spawning bluefin. That was the most heretical thing anybody could say. And believe me, it's still heretical, because 20 and 30 years of biological assumption to some people can be wrong until you basically take a payload of evidence and put it in front of people, and then be prepared to defend it and to keep pushing for the next 10 years. Because bluefin is a tough place. It's a very political, as you can imagine, fishery. So we discovered in the very first steps and days of Atlantic spawning grounds for bluefin tuna And again, this is the early days. We made some predictions based on that. We brought all the pieces of the story together, like history, hormonal analysis, basic studies, and predicted what we should find, and then finally other scientists were able to come and join us and provide the evidence. So now the next steps are, okay, this is just the beginning. And what's really interesting to me is that bluefin tuna may turn out to be like the other tunas in that its spawning areas could be much more broad than we thought. And it's really about, for spawning areas, it's really about larval success. Because as I hope you remember that bluefin can go just about anywhere as based on their physiology. But their larvae have to develop in the best places for their survivorship. And so, if you remember the old view, with separate and exclusive spawning areas and vastly different ages of maturity, we feel that we put enough scientific evidence on the table to show that those are incorrect assumptions. And we're hoping that the first tracks are basically going to lead us to, in technology, better understand, and you can appreciate how difficult it is to get out to the open ocean and study whether fish are in spawning condition or looking for the larvae. It's going to be challenging. So I'm going to finish up with, if you remember the last slide where you saw the spaghetti, basically the conventional tag results that show basically fish crossing the Atlantic. What we showed here, all the colors, are the consolidated tracks of different labs over the past 17 years that show the routes of migration for bluefin tuna. And I'm happy to say that my lab is the blue, the contribution of the blue. And the NOAA and then European scientists are getting better at tagging and keeping tags on. But you have to be able to keep that tag on a year to get a migration cycle. And what we're learning is that even now, some of the fish that are tagged in the West are entering, after spending various time in the West, are entering the Mediterranean. So there's even a question of whether there is such a thing as exclusive spawning site fidelity. I personally predict that there isn't for bluefin, but right now, for 30 years, the assumption is spawning site fidelity. Fish come back to the same place year after year to spawn. If they're actually true for a highly migratory species, it might be wrong over 20 or 30 years because the conditions for their larvae would change. So you can appreciate that along with very unique species like bluefin, might not be too surprising that they wouldn't put all their eggs in one basket and do the same thing year after year. We already know that they shift their migration routes, individuals, over a period of time. But what's really cool to me is that the evidence, cooperative research, and using technology is starting to fill in the gaps of all the unknowns in bluefin. And believe it or not, this is the first year that this kinds of information is being considered at all in stock assessment in Madrid, actually this week. And it took us 17 years to convince stock assessment scientists and our US government to utilize this fishery independent information. So it's a very conservative area of fishery science, stock assessment. So stock assessment scientists, and hopefully you're going to be new people, are going to use new kinds of information. And believe it or not, that's what the old scientists used. They used life history and other things. So here's just a couple of final thoughts for you to think about. The individual bluefin dispersal patterns are far more complex than anyone ever guessed, including fishermen. Here's just one track. It's a year-long track. The stippled area is the error associated with the geolocation estimate. This fish we tagged and released off southwest Nova Scotia, probably in October. It was a big fish, 800 pounds. It hung out down in the southern Caribbean, down here in the southern North Atlantic. Then it ran into the Med during the presumed spawning period, left the Med, and a year later, its tag reported off on the Grand Banks. We don't know what happened on August, though. It's actually August. So, by October, it might have made its way back to where we tagged it. Most of the fish that keep their tag on a year, whether it's a swordfish, a bluefin, a big eye, they're almost always back to where we released it. Don't ask me why. If we had a 3 or 5 year tag, I'm sure we would see that it moved in many cases because we know that they do. So that just shows you what you can learn if you stay on something for 15 years and keep your nose to the grindstone and don't believe everything they told you. And so the citizen science part of the story is pretty cool too. We decided that since there was almost no information on juvenile bluefin, because there's no commercial fishery for them under management regs here, we would try to engage recreational fishermen in tagging, just spaghetti tag. So we called it the Tag a Tiny program. It was sort of a joke. Our competitors at Stanford had the Tag a Giant, and we thought we used to have a tag a real giant, but anyway, I digress. So we engaged fishermen and put a tag on a fish, measure it, and let's see where they go. And unlike the pop-up tag, they're not $4,000, and if they stay in, you can have multi-year recoveries. And I'm just going to show you the most recent data. I'm sorry, if this were interactive, it would animate, but all of the fish were tagged mostly here, some right off here, by sport fishermen for the most part. And the colors indicate how many years at large at Liberty. And you can see anywhere from recent recoveries up to 8 years later. And I want you to see that with 30 or so recoveries, you can't see too many locals. We had a fair number of transatlantic recoveries. And I can tell you that they go both directions. I'm only showing you our recreational fishermen's releases. What I'm not showing you are the scientist releases from the Bay of Biscay and Med and in here that have been recovered by our fishermen and our fleets. So the assumptions that fish crossing each side was less than 1 to 4% a year is totally wrong. We don't think it's climate change or ocean warming. Based on old historical records, it could have been the case forever or for bluefin. We won't know for sure. But the whole point is that a lot of the assumptions used for stock assessment and bluefin biology are not holding up. So it's up to you guys. For all the other species of tuna for which there's far less known in the Atlantic, step up. For the billfish and the bluefin and the other tunas and all the other species, there are huge gaps. And it started with fishermen that said, what we see is not what you say is the status of this stock that we're fishing for, and which, by the way, has been very well rebuilt in the past decade in the Atlantic and is under sustainable fisheries measures in the U.S. There's no endangered species status of bluefin tuna, and the good news is that once the overfishing in the Med was brought down, bluefin tuna in the Atlantic is looking pretty good. And along the years, we were the first, going back to the aquarium, to pioneer these fishery-independent methods using slaughter planes. We attempted UNH, my Seacom buddies, using acoustic methods. We actually got some drones out there. They're finally using aerial surveys in the Mediterranean to assess bluefin, both spawning bluefin or presumed spawning and adults. It's not yet used or supported financially in the U.S., so stay tuned for that. The last couple of things we did for fun, this is the National Geographic Critter Cam. We basically brought my old friend Greg Marshall. Where's Greg? Somewhere over here. He's the inventor of Critter Cam. Actually, no, I'm sorry, you took the picture. That's my collaborator, Tim Lamb. And we were trying to basically get camera gear on bluefin, basically as a means of figuring out what else we could put on the bluefin and see what it was doing. So there's that, there's the Critter Cam, and that's Greg right there. So that was kind of a fun thing to do. And my last message to you, this is the age of fake news, questioning scientists, questioning our expertise. You all are facing things that some of us older scientists didn't face in our careers. We weren't mentored to deal with these things. So we're all in it together. Times have changed, and you scientists are going to have to learn how to cope like we did. We happen to be in a controversial field. I happen to be a bluefin scientist and expert for the last 20-some years. Nonetheless, sometimes there are campaigns that don't necessarily accurately represent the status of maybe the species you're studying. And that's when you have to be ready to stand up and defend bluefin on the brink of extinction, or that all tunas are overfished and down to 2% 2.6% of its biomass. I didn't expect this. I thought if I was a good scientist and my lab did good work, that it wouldn't be a problem. But it's reality. So you have to be prepared to defend. Wherever your science is questioned or misrepresented, be prepared to stand up and fight for it with your colleagues, your collaborators, and your students. And also, be prepared to talk about what your science means. And this happens to be Diners Journal, New York Times, and many chefs throughout the U.S., they think all tunas are endangered, and they won't serve bluefin because they've been told that it's on the brink of extinction. In fact, we have highly regulated and managed U.S. and Canadian fisheries for bluefin tuna. So what is the reason that chefs are told that they shouldn't serve it? You may have a personal belief to not eat animals or meat, but as a fisheries scientist, if we've improved the status of fish and the population and sustainable fishery, why in the world would you tell a restaurateur or consumer not to eat it as far as based on the the population status? So these are the kinds of things you might encounter as a tuna scientist. And I tell them the good news, and they're all so happy. Chef in Seattle, it was an amazing sushi restaurant. He said, "Oh, I'd love to serve bluefin, but I can't. It's on the Monterey Aquarium list of do not." I said, "Let me assure you, you want some papers? Here's the U.S. quota, and you can actually affect in your conversation portraying science, affect economics, affect seafood consumers, and so on and so forth." So I guess I hope you learned. By the way, this is Lizzie Jagger. That's Mick Jagger's daughter. Okay, I covered her up. I know you're all adults. She was nude. This is the Oceanic campaign. This is the yellowfin tuna that they were portraying as the endangered bluefin tuna that you should not eat. Over here is Linda Greenlaw, your neighbor up the coast, who is commercially fishing for swordfish. And I just wanted to point out that complex fisheries and science issues require good science, honest advocates, and ocean tools and new skill sets that you will most undoubtedly have that we didn't have in my cohort. So get to it. And then we're also up against the whole question of impacts of ocean warming and climate change. If we don't have a baseline on these animals, how are we going to know the effect of ocean warming? So another reason why we're way behind the curve. And so we need creative, science-based, interdisciplinary approaches. Work with engineers, work with the techies, work with economists, work with fishermen to get the answers of hard questions. So I want to thank so many people that contributed to the information I presented to you. I hope you get that a lot of this work was conducted with fishermen who brought the questions to us, helped us develop our tools, and had a stake in it, and their families and their communities. And Noah, Guy Harvey, our own funding, by the way, that's Scotty Durbinowitz. He was the highliner on the sword, Life on the Line, Dudley's Catch. And so funding is important, and finally, if you want to see anything else about our other species that we work on, tumalab.org is our website. So thanks for your attention, and I'm happy to answer any questions.
So you mentioned that the previous population estimate was 7,000.
That was 92.7 plus with error.
And so what did you find with your surveys?
Our surveys, we counted up to, the range at the time was, and again, these are minimum surface counts of a three-dimensional school, so we only count the top. And so upwards of 150,000 adult, with 73 inches and above in the Gulf of Maine alone. And remember that bluefin occur up and down the coast, all the way from the Gulf of Mexico all the way up to the Gulf of St. Lawrence. So at the time, but none of that information was ever used in any assessment because they said it was absolutely not comparable. And it wasn't even used as a ground truth. It was used politically. If anything, when congressional staffers were in management and advisory hearing, the staffers, I wasn't even allowed to speak. And frankly, it was early days. But when they showed that picture of a school of 5,000 plus, one school, that said a lot. And that political information that then stayed, there was a proposed listing of bluefin tuna by National Audubon in Sweden as an endangered species. And based on the assumption that there were only that many left, which was wrong, and that prevented the 50% quota reduction. None of the aerial survey information has been used directly in any regional assessment of abundance, even though that was our goal. It did not receive support because they weren't ready. But now, 20 years later, in the Med, ICAT is using aerial surveys for the first time. But they're not using quantitative methods. They're not using sonar or drones or any size verification, which is what we were trying to do with our drones and our sonar to get the total biomass with the known sizes of the individuals in the school. The bar was set pretty high. We didn't get there, and then the funding went away.
So can I just follow up? So
Sure.
I don't know what the fishery dependent estimates were, but is it possible that they're still accurate in terms of estimating the percent of the original population? Right?
Yeah, I personally, I think from all our work, I think it's almost what's the word? I think the assessments in the Med are more useful because the heavy overfishing, the vast numbers of fish that are taken out. But I personally think that that the very low quotas in the West, and basically you can't count what you don't catch. If you hold the catch to specific levels year after year, you will never know what you didn't catch. I personally think that we were just managing, basically we were observing that the catches and the indices of abundance were fairly consistent. But I don't think it ever truly gave us a handle on what the absolute abundance was. And now the whole thing is blown out because now we know that the degree of mixing is probably so high, and we may not even have spawning site fidelity, that even though there might be subpopulations or metapopulation, that means a lot more diversity in the stock, if they're mixing so great, maybe all along it should have been assumed that it's one Atlantic-wide population. So I personally, I know people, stock assessment scientists and statisticians may not agree with me, but I think Pacific fisheries tuna scientists also agree that sometimes you're not even touching reality. You're just really monitoring catches really well if you're not overfishing. If you're overfishing, it's a whole other story. And I think it's based on, we didn't know spawning grounds. We were off by age of maturity possibly by as much as 5 or 6 years. And in our PNAS paper, the point we make, those fish aren't even counted anywhere, and there's no way to measure within the approach to tuna stock assessment is a very old one, virtual population analysis. And that cannot account for productivity, and it doesn't include the spawning stock biomass from production. So I'm sorry I'm getting into the weeds, but what I'm saying is I don't think we ever knew how many bluefin are out there because the way they're counted doesn't have anything to do with the fluxes in the population in the Western Atlantic, because we couldn't monitor it.
Is it possible you're both right that there was 1.5 million before, and now there's 150,000?
Oh no, 150,000 is just the fish we counted at the surface. If we were to extrapolate from that, we would have to go up to orders of magnitude more fish because the three-dimensional schools, that's a vast minimum and didn't include any other bluefin in any other part of the Western Atlantic. So that's a regional minimum count of how many fish were here. So 150,000 fish is like how many people are on this island right now compared to how many are in New England. I don't think it had anything to do with the population. All we said is, this is what we counted, and you said that the stock could be as small as much less than that. And that can't be because here we can show you their pictures. But it just shows the uncertainty that's involved in the supplemental assessment process.
So what did genetic analysis say about the whole eastern versus western?
Yeah, believe it or not, after all the money and all the attention and all the sampling, this is, I think, the first year where and it's not even published yet, but Astotechnalia appears to be able to distinguish substock. And so far, all they could resolve were eastern and western. And again, the assumptions were that western were all Gulf of Mexico fish and eastern were all Ned. And now we know that there's spawning grounds in the Atlantic. And now we know that there's much more mixing. But we're looking really forward to see what the latest results are. The other techniques, say microconstituents, I don't know if anyone ever talked about that, chemical signals. The challenge there was the assumptions were, again, the same, that there's only 2 spawning grounds and you either had to come from either place. And then all the evaluation of the chemical signatures were all based on that assumption. And most of the samples actually were unclassified. There was a middle ground. Well, that's probably because there was a middle ground in spawning areas, and there might even be multiple spawning. One fish might have spawned in different locations, like other tunas, over their lifespan. Why bluefin got to be the magical tuna, I don't know. They should have, if we had Pacific tuna scientists that studied Big Island, it would probably not have held for so long as it did, these assumptions. Very few people study bluefin tuna. As they say, it was too political, and if you couldn't stand the heat, stay out of the kitchen. No one wanted to study bluefin tuna until money and power came in. And we studied it because we started small, a $25,000 grant, and we just stayed at it. And Stanford has been in it big time for a very long time, with much bigger money. And what's interesting is the stories all converge. We just interpret them differently. The fish we still get the same answers, but we interpret them differently. The results.
Do we know spawning seasons of tuna? Like, do they have a certain, like, if they're seen in this place in this month, they're most likely spawning kind of information, or?
Yeah, in the Med, bluefin has been treasured and prized since before Christ. Aristotle coined bluefin tuna in the Med are the most studied and known creatures. And there's a phenomenal amount of information on spawning food. But believe it or not, it was assumed until probably the 90s that bluefin only spawned near the Balearic Islands, because that's the only place scientists looked for them spawning. Even though fishermen knew that they had gonads and other things. So first in the Med, it was shown that, oh, wait a minute, if we look for larvae, we're going to find them. They found larvae almost everywhere they looked, but the spawning period in the Med, it starts in the east, and the smallest fish spawn first near Turkey, and as there's bigger fish spawn later in other parts of the Med. So it starts in spring, and it finishes up pretty much by August, when we know that bluefin leave the Med, at least with the adults do, and there's even now new evidence that bluefin that used to spawn in the Med may be spawning off Africa, and may have been all along, just that no one documented it. In the west, the Gulf of Mexico, the known spawning period is April and May, and almost all the tigefish are out of there by June. When the ocean is just too hot, it's 80-some degrees, it's too hot for larval development. But in our paper in PNAS, we found larvae as late as September, up close to the Grand Banks. So as we predict, because the warming Atlantic Ocean and the long days allow larval development at higher latitudes. And so I was actually surprised. But the only reason why it wasn't a total shock when they found the larvae on the Grand Banks in, say, September was that Gilad Heinisch, my PhD student, when we sampled the hormones of fish in Nova Scotia in September and October, he said, "Moly, he's got a really emphatic voice, moly, these fish had spawned within a month." He said their hormonal profiles showed clear evidence that they had spawned recently. And I was like, oh my goodness, Gilad. That means they had to have spawned close to here, because we have all the tracks. And he was right. Even though our sample size for hormones was small, it was 10 a year, he was right, we think, because the evidence was the larvae. So there's a lot more to do with that. And going back to me it looks like there's a continuum of spawning from the Gulf of Mexico all the way up to the higher latitudes, just like Pacific bluefin. Start at low latitudes and work their way up in the Western Pacific. And it appears that may be what's going on for Atlantic bluefin in the Atlantic. And but it's early days. detection tag. We've been working on that for years now. That tag won't do it. We can only infer that it might be spawning. We need physiological evidence of spawning, or we need absolute behavioral confirmation, say an accelerometer tag, that you use that accelerometry data and confirm the actual physiological evidence of spawning. Once you do that, then we'll know where spawning zones are. And not just for bluefin, but for all the tunas and the billfish, which for the most part remain unknown in all oceans, which is shocking.
So I have two questions, and they're related to your last statement. What's the length of the larval phase, and how much distribution occurs during that phase?
Yeah, they grow really fast, about 1 millimeter a day, and that's the first, question that we had from some of the, questions about the veracity of our paper and could all those larvae been infected from the Gulf of Mexico, and very convincingly showed there was no way, because if 1 millimeter a day, we were finding 3 and 5 millimeter larvae. And so, but you're right, they are distributed in currents, but they really love in circulation areas, as you know, we think of the spawning grounds, so they're not affected too much, because then they wouldn't get food if you're a larvae guy, you might know that they need the larval habitats, have to be not too many predators, not too many conspecifics where they eat each other, but they need enough food, the right kind of food to survive. And those areas were predicted by the Sutton model.
And in that model, is it repeatable year to year, or does it vary a lot?
It varies with the input in, yeah, and then if it can go forward and go back, we wanted to 50 years back. And so in the Indian Ocean, we've been looking at it, and they're actually doing end-to-end modeling with stock assessments now. To see, but the whole point is to bring biophysical information that might govern productivity in early life history. I mean, some of us think that the real drivers of the stocks, the fluctuations in early life history, that maybe what's important is how well the year classes do with the larval year classes, and I think that's been shown for many other fish species, but it's been so hard to do in these pelagics that I don't think it's going to take a while. But you might be able to do it with modeling, hindcasting, and forecasting. But you can appreciate how hard it is.
Absolutely. All right, thank you.
Well, I personally pondered living at seabirds and living under the tuna right now. Thank you so much for that inspirational talk. And let's just thank Mom for being here tonight.
Have a great visit.